Hello, my friends, and welcome to our tutorial on meiosis phases. Oh, always do that. Phases. And we have a special guest, special guests today. We are going to welcome our friends. So let's have a little welcome for our guests in the studio today. Very nice. We have the Californians here. So that is a little bonus for us. And we're going to talk about the meiosis phases. We're going to go through this very quickly because I know you guys can read and you have a book, and so you should use it. So, right, interphase is going to always, always, always come before mitosis or meiosis. It's where we get replication, right? And so we get sticks into X's for all of our chromosomes. So we get a sister chromatid, an identical piece of DNA, right? During interphase, before anything else starts, the big picture, meiosis I, separation of homologous chromosome pairs, reduction of number by half. What does that mean? Yes, my friends, we are haploid after meiosis I. And then what happens in meiosis II? Meiosis II is essentially mitosis. It's the separation of the sister chromatids, the equational division. So what happens? Yes, the gametes are haploid at that stage too. Continue to be haploid. All right, so what happens in meiosis I? Prophase I, what is synapsis, right? Pair up of the homologous chromosomes. That's the most important thing that happens in prophase I of meiosis. What's a tetrad? That's when the four sister chromatids of the two homologous chromosomes get together. So we always draw them next to each other, but they really could just kind of just be on top of each other so that each of the sisters has a non-sister to pair with. And then that's when chromosome crossing over happens. It is a physical exchange of the chromosomes. What do you mean, what am I doing here? I'm here trying to teach something. So I don't know what that really means. You're the jerk move, Stuart. Well, who is Stuart? And I didn't do a jerk move. I'm just trying to teach some meiosis. Honestly, people. All right, moving on. Prophase I, pairing of homologous chromosomes, crossing over. Here's some nice pictures, right? Here's all the little stages that we can talk about for each. Still talking about prophase I. Remember this guy, the singular version, Kismata, where we actually have the physical exchange, that cross part. And then here, finally at diakinesis is when we actually are down right into our sausage-shaped chromosomes. These are all spaghetti, right? They're working on getting condensed as we go through here, but we're not actually condensed.
Devin? Brad? What just is going on here? I don't know, Stuart. I'm not confused.
What? I don't really understand what you're confused about. It's just prophase I. It's just pairing of homologous chromosomes crossing over between non-sisters. It's not so bad. Okay, so metaphase I, align the homologous pairs along the metaphase plate, right? Anaphase I, separate the homologous chromosome pairs, random distribution. This is where we get the whole independent assortment, independent assort, woohoo! And then telophase I, right? They're at the poles. It's they kind of decondense. They don't look exactly like sausages there, but it also could be just because they're jammed together, and that's what we can see under the microscope. Interkinesis is just cytokinesis, but they call it interkinesis because it's not done yet. We're not done. It's just meiosis I. We're not done. All right, so we're going to do a little what happens during these, and then you're going to do a screenshot and upload to number 7. So just write it out. It's a fill in the blank. Super easy, super simple. Take a screenshot, upload to number 7 in meiosis for tutorial phases. Tutorial phases in Canvas. So go ahead, take a pause here and make that happen. I got to take a break. Hold on. We have some issues happening right here. All right, I am back. Sorry about that little delay. Where were we?
I saw Darth Vader.
That is great. I'm glad you saw Darth Vader, but what does that have to do with meiosis? Nothing. Okay, moving on. Oh, pictures. Pictures are always fun. So make sure you know the steps, right? Here you go. Here's all the nice little pictures with what happens in each stage. Meiosis I and meiosis II. Prophase II. What do you think happens? Not a lot. Condenses back down if they weren't already condensed. What about metaphase II? Line up on the metaphase plate down the middle. Interphase II, pull apart. Telophase, right? We're getting everything to come back decondensed. We're getting the nuclear membrane around them again. And then bingo, after the last cytokinesis, there we go, 4 cells. Riveting, I know. Oh, now we're not going to go through showing all these up front. You guys can just look at those. Oh boy, yeah, there's like a million slides of these, so we don't need to read these to you. Boink. Yeah, all the way at the end. So what do we get? 4 cells from each original cell. Chromosome number in each new cell is reduced by half. New cells are haploid.
Brad? What did I just see going on here? I don't know, Stuart. I'm not confused.
I'm sorry you're confused. I don't know how it's confusing. I'm trying to be as clear as possible, right? Haploid, half the number. Meiosis I, right? We already have half. We pulled the homologous chromosomes apart. Yeah, they each have a sister, but they're connected at that centromere. So if they're connected, they're still called one chromosome. In the meiosis II, right? The sisters come apart, but then like they're still the same number, even though now they're split in half, but there were 2 because they were connected and now there's 1. So it's haploid, right? The only place that it's diploid during meiosis is before we go through meiosis I. Obviously interphase. And then like what, prophase I, metaphase I, anaphase I technically, because it's not interkinesis. Telophase I technically, once the interkinesis, then there's 2 cells with half. Haploid. Go! Here we go again. Here's another, fill in the blank. Upload number 8 to today's tutorial, meiosis, phases. Upload number 8. In Canvas. You guys love Canvas. We all love Canvas. All right, take a break. Go ahead and do that. This is just another little picture, right? This is exactly what we talked about before. You can see, right? Non-sister chromatids exchange pieces. So if crossing over is happening, what phase does this have to be? Yes, my little friends, prophase I. Yep. It's just prophase I. And then 1 and 2 happen. And then look, we get 4 at the very end, 4 gametes, each are haploid. Shows you how we have some recombination, right? Originally it was big A, big B, or little a, little b, but some of the gametes, oh look, it's big A, little b. Ah! That is crossing over. Okay, come to office hours, Californians, and I will help explain it a little further, but we need to move on. This tutorial is already getting too long. What else? Oh, now we have 3 chromosomes to look at so they can show us all the different combinations, right? It's just an example. Different ways they can align at metaphase I determines, right, what happens. Now we're not talking about, they're not even adding in crossing over right here. They're just talking about what happens to the chromosomes, right? So what is this, 2 times? Because there's 2 of them for each, because you get 4, right? They're only showing 2, don't get crazy. There's 2 of this one and 2 of this one, that equals 4. Okay. Okay. We'll keep talking. It's going to be fine. Go. We can't really see it because it says meiosis upload number 8. Fascinating. Under tutorial, meiosis, phases in Canvas. Just pick one. If you have an iPad, you can just circle whichever one you think is right, screenshot and upload. If you don't, just write the letter on a piece of paper. A, take a picture of it, upload. B, take a picture of it, upload. C, take a picture, whichever one you choose, and just upload that photo. Okay? Yes? I told you, I'm trying to teach. Apparently, I'm not doing a good job because Californians are super confused. Okay, this is it. This is the last one. This is just it all in one fell swoop. If you know this, then you're going to be fine. Remember, you're going to have to draw it like we did for some of our other uploads. And we're going to draw it on the exam in some form. Not sure how I'm going to make that happen, but you guys are going to have to draw out meiosis. I'm going to give you some parameters and you guys are going to have to figure it out and make sure you draw it. Lots of practice and you'll totally be fine. So I'm not worried. It's all going to be good.
Brett?
What?
What did I just see going on here? I don't know, Stuart. I'm confused.
So I guess the Californians are still confused. There's not a lot I could do about it. So thanks so much for watching. And I hope you guys are less confused. If you're confused, just please shoot me an email. Make sure you go to the PLF. It will totally help you. Appreciate your time, guys. Thanks so much.